let's go ahead and get started. Thank you again for joining us for the Southern IPM Hour. This one is on the epidemiology and management of cotton leafroll dwarf virus, an emerging insect-transmitted virus in the southern United States. This is a Southern IPM Center where we present research issues and programs in integrated pest management from the Southern region through this webinar series. I'm Kayla Watson. I'm the Communication Director of the Southern IPM Center, and we are housed at both NC State and UGA with a mission to coordinate IPM across the region. We're also one of 4 regional IPM centers supported by the USDA NIFA Crop Protection and Pest Management Regional Coordination Program. Just to let you know too, the RFA is out for the CPPM program for ARDP project proposals. So if you have a project like this that you'd like to do, those proposals are due on February 15th. So you've got a little over a week to get those in if you'd like to do that. And as I mentioned, this project is an ARDP or Applied Research and Development project, a part of the CPPM program at USDA NIFA. This talk will also be recorded, and that link will be available after the webinar. If you have any questions for the speaker as we go throughout the talk, we will answer those questions at the end. So you can put those into the Q&A box at any time, and we'll be sure to ask those at the end of the presentation. But our speaker for today is Alana Jacobson at Auburn University. And Alana, thank you so much for sharing about your project today.
Great, thank you for having me, and thank you everybody for attending. So today I'm going to talk about research that my lab has been conducting on basically investigating a new aphid-transmitted cotton virus, cotton leafroll dwarf virus. I'm going to start off my talk just saying my thank yous. I'm going to be presenting research from a CPPM project that was funded by USDA NIFA, and matching funds for this project were supplied by Cotton Incorporated. Some of the background information I'll be presenting today and other research also came from USDA funding and also funding from the Foundation for Food and Agricultural Research. We've had a lot of help with regional collaborations from entomologists, plant pathologists, and agronomists across the Southeast region, and my lab members, who you will see pictures of as their projects come up, have also been a critical part of getting this research done. So the problem we're going to talk about today is cotton leafroll dwarf virus, which was a new cotton virus that was first identified from samples that were collected in 2016 from Alabama. During this year, there were a lot of things going on. Crop consultants were seeing some weird late season symptoms, and there's some pictures of some of the common symptoms up in the right-hand corner. But at first, people were thinking, well, maybe it's some dicamba drift because that was also a problem that year. But it just didn't seem like that was the sole reason for everything that they were seeing. So Judy Brown came and collected samples. Our nematologist at Auburn University, Kathy Lawrence, helped her to get samples. Judy is a virologist at the University of Arizona with expertise in identifying different viruses, and she had worked with cotton leaf crumple virus in the Southwest, which was a suspect virus. It took them a couple of rounds of sequencing because they started looking at DNA viruses. Then they did some RNA-seq, and they identified this virus as cotton leafroll virus. And what we know about this virus is it's from genus Polerovirus, family Luteoviridae, and it is transmitted by the cotton aphid. This has been a really challenging problem to work on, partially because symptoms are highly variable. There's currently no well-defined symptomatology for this virus. And if you go out and test plants, the ones that test positive can exhibit any number of different symptoms, from leaf curling or cupping, reddening of veins, leaves, stems, or petioles. Sometimes you get these shortened internodes and like elongated top growth. Some plants might appear stunted early in the season. There seems to be reduced bole set in some of the more severely diseased plants, but not all plants exhibit these symptoms. And there's also a good number of asymptomatic infections occur where you look at a plant It tests positive, but there's no apparent symptom on the plant. So it's been really challenging and remains challenging to diagnose because of the lack of clear symptomatology associated with infection. The other thing I wanted to say, so there could be a number of different factors that maybe are responsible for the different symptoms that do appear. We still don't have a good handle on how they appear in different genetic backgrounds of cotton, but we have detected cotton leafroll dwarf virus in all commercially grown varieties in the United States. The next thing I wanted to show, if it'll play, is a video that one of our extension agents took.
Field in Auburn County showing nearly 100% cotton leaf roll. You can see this is November 16th. There's plenty of vegetative growth plants, flowering all set in the top of plants, but there's plenty of square species.
And so I don't know if you guys can hear the video well, but that was Austin Hagen talking about this particular field. The video was taken in November, and what should stand out to you is this field hasn't been harvested. It actually didn't have any bolls set on the plants, and the entire field looked that way. And so although we don't, the current, if you fast forward from 2017 to now, people don't perceive cotton leafroll dwarf virus as a problem because we haven't had many problem fields like this since then. But this showed up early on after the identification of that first sample of cotton leafroll dwarf virus, and this certainly got our attention and got us concerned about looking at cotton leafroll dwarf virus. The other thing I wanted to say about this virus is after the identification, so other parts of the world where this virus has been found include South America, I think Timor-Leste. It's been reported from India and chickpea, a sample of, I think, aphids. They actually identified the virus from in China. All of the red stars, these were locations where there's actually been some sort of a PCR confirmation. The disease was originally reported from Africa, but those have not been confirmed with DNA-based methods. The other thing is, if you look at the phylogenetic tree on the right-hand side of the slide, basically the samples that we found in the United States are distinct, but they appear to be closely related to samples that had been collected in South America. The current distribution of cotton leafroll dwarf virus in the United States is shown in this map. I think this map will probably be revised shortly. There was a presentation at the Beltwide Cotton Conferences this year in January where they actually found, I believe, samples from Arizona, and I can't remember which other state in the Southwest, but it's likely that this virus is distributed across the cotton belt. We still don't have a good handle on differences in, I guess, incidence and abundance of this virus in these different regions, but we're in the process of conducting surveys to look at that currently. Okay, so before I get into some of the biology, ecology, and research questions that we were looking at with this project. So where we started is we knew this was an aphid-transmitted virus. We knew we could have fields where there's no apparent loss, but we had that bad problem field where there was 100% boll loss across the entire field. Other severely infected fields didn't necessarily have 100% of the plants without bolls on them, but it wasn't uncommon, or there were problem fields where we saw plants that had a reduced boll set. So this plant, you can see there's 2 cotton bolls open, but there's really no other bolls in other parts of the plant. And so a main goal of my research program was to try and better understand what management approaches that we could take to managing this disease, and also just better understand like how much of a problem is it really causing, and identify risk factors for plants becoming infected, but then also try to tease apart like what is actually causing these yield loss symptoms versus the plants that remain asymptomatic and apparently healthy. And so this requires us looking at a combination of different factors. My background is in entomology, so I do a lot of work with vector-virus interactions. But basically, for this disease, or disease caused by this virus to manifest, you have to have the vector transmitting the virus into host plants. And then there's also going to be an environmental component that regulates the vector population. It can influence virus infection and persistence in the plants and the symptoms that develop. There was work done at UGA that identified, I believe it was 23 different weed species. And if you look through this list, you should recognize many of them because they're common in southeastern agricultural research or in agricultural systems. And basically what this tells us is cotton leafroll dwarf virus has a number of very common hosts, and it's probably well established in the landscape. With respect to some background on the vector, the only reported vector for this virus in cotton is the cotton aphid. And if you're not familiar with aphids, aphids basically go through winged and wingless life stages. Basically, the winged stages will disperse, find host plants, and begin reproduction. After this, their offspring are usually wingless, and basically diverting resources and energy from wing production to reproduction helps them expand their populations very quickly. When the populations become crowded or host plant quality begins to decline, they then start producing winged morphs again, and they can repeat this cycle over and over. They have a very high fecundity, so one female can give birth to 80 offspring, and so the population doubling time for aphids is very quick, and so they can grow to large sizes very quickly. What we knew about, or what we know about transmission of cotton leafroll dwarf virus from cotton aphid comes from a couple of studies. There was a study done in Brazil that had a partial characterization of the the mode of transmission. And then we had a student, Bree, who also repeated this with US populations of cotton aphid and local CLRDV strains that were collected. But basically what you can see is the time it takes for an aphid to acquire the virus during feeding is very short. So this can acquire in as little as 30 minutes. When that aphid becomes virulliferous, so it will ingest the virus must then circulate through the insect to be transmitted again. So it'll move down to the midgut or hindgut. It moves across those membranes into the hemocell, and then it will circulate in the aphid's body cavity until it can contact the salivary glands, enter the salivary ducts, and be transmitted again. Once this aphid is capable of transmitting the virus, it only takes 15 minutes to 30 minutes of feeding to actually transmit that virus. And the winged forms seem to have an ability to transmit a little bit more quickly than the wingless forms. The other thing we looked at was once an aphid can transmit the virus, for how long is that aphid going to transmit? And we did this by looking at successive transmission over different days during the aphid's lifetime. And basically what we saw is the aphids can transmit the virus for up to 15 days for the wingless aphids that we tested and 23 days for the winged. So basically once they've acquired the virus, they can transmit for the majority or the rest of their lifetime. Something else to consider in this scenario when we started research is cotton aphid is the primary aphid species that annually colonizes cotton in the southeastern United States. It is the vector that we know transmits CLRDV to cotton. But there are 7 other species of aphids that can occasionally be observed to colonize cotton. These are not pests of cotton. We don't manage them. We don't even really look or scout for them. But every now and again, if you're in a cotton field, you may see any of these species. Two of them, the Cowpea aphid and the green peach aphid, were particularly interesting because in India, where cotton leafroll dwarf virus can infect chickpea, these two species were reported as vectors that transmitted the virus to chickpea. We tested them for transmission in the United States, and we did not observe transmission to cotton. But I don't know if we can completely rule them out as vectors because mature cotton plants are not good hosts of these aphids. The populations really struggled to persist. So I think it's safe to say they probably don't play a large role in spreading the virus to cotton, but they might be acquiring and transmitting in the landscape among the other weedy hosts. We still don't know the vector status of these other 7 species, but they are in the system. We know cotton leafroll dwarf virus can be transmitted very quickly, so it's not unreasonable to at least consider or want to understand more about these species so that we can rule out whether or not we think they might be playing a role. So before I get into research objectives and talking about some of the projects that we started to try and look at spread and management, this is kind of showing a diagram of what typical virus spread looks like seasonally in the southeastern United States. And this is kind of based on what we know about other insect-transmitted pathosystems. And so you'll have a crop growing season, and here we have cotton. We'll usually start planting in May, harvest October, November, December, end of the year. The aphid populations will be on alternate hosts in the landscape that are also hosts of CLRDV, or some of them will be. And so these are just a variety of different winter, annual, perennial, and then there will be summer annual plants that come into the landscape a little bit later. And typically what happens is when the insects need to disperse from these overwintering hosts and find new hosts, that's when they end up moving into the crop. Now, with respect to when we typically manage cotton aphids, we usually manage them, or we see their populations in cotton in June and July. And they'll usually show up late June. Populations will establish on the crop. They build to high numbers, and then they'll crash because of a fungal epizootic, and I'll show you that later. But that's when we would assume that primary spread is occurring to the cotton crop. Other questions we had, though, is, so is this the only time CLRDV is being transmitted? If there are other aphid vectors responsible for spread, it could happen at other times of the year. The other thing is are these populations persisting, and is there additional spread occurring throughout the cropping season? And we didn't have any answers to these questions, and we were interested in figuring that out because when the virus is being spread and how many times tells us how many potential interventions we might need to protect our crop from virus spread. So our main research objectives early on, after we detected this problem, were to get a better understanding of these aphid population dynamics. And this is both for the known vector, the cotton aphid, and also these other 7 aphid species. And to better understand when CLRDV spread is actually occurring to cotton. The other thing, of course, we wanted to evaluate are different management strategies. So this could be vector management, cultural practices, host plant resistance, but what can we do to reduce disease severity and loss? And there was also an educational component to this program where any of the results that we have generated from the research programs, we have handed off to our extension counterparts and disseminated to stakeholders of interest. So I'm going to kind of talk about this with respect to the order in which we did projects. And the first project that we really wanted to look at is cotton aphid management and whether or not that could reduce virus spread. And that's just a logical first step, because one thing we were concerned about is if you tell a grower we have a new cotton virus and it's transmitted by our aphids, the first thing they're going to think of doing is, well, can we kill the aphids and then eliminate the problem? And this is something that we wanted to understand whether or not this was actually a viable strategy, because for many pathosystems, you can't use insecticides to reduce virus spread. And that's not true in all systems, but it's not a universal rule that that's a good idea. And we didn't want to increase the chemical footprint if there was no benefit. So the other thing is, although there are some growers who will manage for cotton aphid, a lot of growers, we don't recommend management actions. And that's again because you have this fungal epizootic that will come in and the populations will naturally crash. And I have a colleague, Philip Roberts at University of Georgia, who has looked at this. Basically between 1998 and 2008, he conducted 27 trials where he looked to see whether or not there was a benefit to killing aphids just from a yield loss perspective. So this is before the virus came here. And basically you can see that there's no difference in yield between treated and untreated plots. And so really the only times that we are recommending growers spray for aphids is if this fungal epizootic doesn't come in. Sometimes that can happen on like a hot, dry year. Or if you have a late planted crop and you get heavy infestations when they're seedlings. And again, that's probably more from a, you don't want to delay maturity standpoint, not necessarily a yield loss perspective. So we were concerned about the effect that insecticides might have on this whole system. So I had a graduate student, John Mayhoff, who looked at this for one of his studies. And we tried to design a series of treatments based on what a grower might currently do versus what insecticide interventions might do to reducing virus spread in the crop. So we had our untreated control. We had a treatment where we tried to prevent colonization of cotton by the aphids. And this was done by basically trying to eliminate aphids from the systems by spraying the crop every week before the aphids come in and infest the crop in June, all the way through until late July, after populations had crashed. Treatment 2 was to spray at the first detection. So the aphids would colonize the crop. In theory, primary spread, so transmission could occur, but then you could kill those aphids and maybe that would limit any subsequent spread that might occur if you let the populations build. Treatment 3 was just kind of another check. That would have been a timing that's consistent with when growers would spray for aphids if they were going to. And then we also wanted to look at the effect of planting date on CLRDV incidence to see if maybe mature plant resistance would have an effect. And I'm only going to show you a couple of slides from this, kind of focused on the take-home messages. And so the first take-home message I want to show you is, so all of these lines, like there's 8 lines, that's our 4 different insecticide treatments and our 2 planting dates. But what you'll notice is if you look at how many plants were infested, we had 100% of the plants infested, regardless of whether you sprayed not at all or if you sprayed every single week for 6 weeks to try to exclude aphids. So we were unsuccessful at excluding aphids in any of these treatments. The other thing I wanted to show you was final incidence of CLRDV in these plots. And so basically in September, we would take a sample of 10 random plants per plot so that we could calculate incidence of the virus. And we had 2 locations, one was in Alabama, one was in Georgia. And at both locations, we had very high incidence of CLRDV. So 69 to 89% in Alabama. We had 95 to 100% incidence in Georgia. And if you work on any type of virus, like you'll recognize that this is very high. and there's no significant difference in virus or the final virus incidence, regardless of what treatment we used. So these results were really important because basically it shows that spraying insecticides for the vector are not going to be an effective management strategy. And that's good to know because we need to make sure that growers aren't increasing the chemical footprint of IPM in cotton, and especially because not only does that increase the cost of the insecticide application, but in both trials, in our weekly insecticide sprays, we ended up flaring all kinds of secondary pests. We have some really dynamic systems in the Southeast, and if you overspray, you create a lot of problems for yourself just because we do have a lot of benefits from natural enemies in these systems. So another thing that we started early on is to better understand the population dynamics of these aphids. Again, I had lots of people working on this, an army of undergrads going out and helping with trapping studies and identification, and several technicians in the lab that helped coordinate things and identify lots of aphids. One of the studies that we did was a collaboration with other researchers across the Southeast and into Texas. So everywhere you see a star, there was a trapping location over a 2-year period. And we basically put up these yellow pan traps around cotton fields to monitor for the presence of aphids. So when we were identifying aphids from these traps, we only focused on the 8 species that are reported from cotton. And so those are the results that I'm showing you right now. And I just kind of wanted to show you an overview. First, we didn't collect any black bean aphids or bean root aphids at any location. The other thing I wanted to show you is, so the orange color in the graph represents the corn root aphid. And this was the most abundant species at all of our locations, and you'll see that on the next slide. The second most abundant species was cotton aphid. The rest of the species basically comprised less than 4% of the total trap captures across all locations. On this slide, I wanted to kind of show you what that looked like across the whole area. I'm just showing you the pie charts with the total number of aphids captured at each location. Again, you can see that corn root aphid was the most abundant species, except for, I think, this one Georgia location and this Florida location. And you can see that the cotton aphid is also second most abundant species at most of these locations. But what I'm not showing, just for the sake of time, because this is only supposed to be like a 40-minute presentation, we also broke this down by the different weeks of trap captures, and the timing of those aphid flights, of course, it's not surprising, but is variable across these regions. And I also just wanted to point out that in areas like Central and South Alabama, South Georgia, where we have high incidence of CLRDV, as you saw in the previous study that I talked about with the insecticide trial, we also have more consistently, we capture more cotton aphids at these locations. So that kind of gives us an idea that cotton aphid is very abundant in these systems, but also it's curious that the corn root aphid is also is the predominant species across this whole area. The other thing that we layered into this, and we were only able to look at this in Alabama because it's cost prohibitive, virus testing for CLRDV remains very high, but at the 3 Alabama locations, we were also able to look at CLRDV spread in the same study where we were trapping aphids. So we basically set up our pan traps around cotton fields to capture aphids, And then in addition to that, we would put out a cotton sentinel plant. And this was a 2 to 3 leaf cotton seedling that was healthy, had been grown in growth chambers, free of pests. And when we set up our traps, we would put out a sentinel plant. And then when we'd pick up our traps, we'd collect the aphids, change the solution, and we'd replace this plant. The sentinel plants would then go back to the greenhouse, and we would spray them with insecticide, get off any insects, and we would maintain them insect-free for 8 weeks and then test to see if they were positive for CLRDV. I also wanted to say that we did this around both cotton fields, and we chose some sort of like a natural area at the research stations we were working at. And we did this just so we knew that if we were getting positive sentinel plants that were infected with the virus, we could say that that was reflective of aphids moving through the landscape and not just aphids that were hopping off of the cotton that our traps were around onto those plants. This next graph I'm going to show you, let me just explain real quick. So we have the 3 locations, South Alabama, Central Alabama, North Alabama. This is the estimated total number of Aphis scassipii. Notice that the y-axes are different scales. So the peaks that you see across the 3 are not the same numbers. And the numbers at the bottom are hard to see, but this is basically beginning March of 2020 and going through that whole 2-year period. The first thing I wanted to highlight was in these boxes, this is the typical time where we have these cotton aphids infesting commercial fields where we notice their populations. And so this is happening in June, and you can see in these graphs a little bit more clearly that these populations build and then they crash due to that epizootic fungus. The interesting thing that we learned from this study is that these populations persist. There's a little bit of a gap here because we had Hurricane Sally taking out our traps that first year in South Alabama. But if you look in both years, we're continuing to capture cotton aphids and through November, so through the end of the year. These little asterisks are also important because these indicate the weeks that we detected CLRDV infection in our sentinel plants. And so you can see that when cotton aphids are present, we get a lot of virus spread. And in Central and South Alabama, virus spread was detected from 13 to 18 weeks. In North Alabama, we saw a very different picture where we only had 3 weeks of virus spread each year. The other thing that was interesting is early season, the first year of trapping, we see the CLRDV spread occurring, but there are 0 cotton aphids being captured in our traps. And I'm not going to show you the data from all of the species, but if you look at the most abundant species during that time, early season, it's this corn root aphid. And this doesn't mean that this corn root aphid is a vector, but it suggests that we should probably test this species for transmission in the future to determine whether or not it is. So from this, we learned a lot about cotton aphid dynamics. Honestly, after those populations crash in July, they're out of sight and they're out of mind. People aren't looking for them, but they're in the systems. They continue to move through cotton and they're continuing to spread the virus. That said, cotton aphid population dynamics and the number of weeks that virus is spread is different among different production regions. You know, we only had 3 weeks of virus spread in North Alabama, so not all areas have the same risk for virus infection. We also suggest that corn root aphid might be an additional vector that could be tested, but this information is really important. It reinforces why we didn't see aphid management controlling or reducing final incidence of the virus. It's highly unlikely that any insecticide spray is going to be effective when you have up to 13 weeks of virus spread each season. And again, just reinforces that aphid management isn't going to be an effective management component. something I wanted to mention, just in case you're interested in this topic and following what we learn in the future. The work from that aphid study that I just presented and the weeks of virus spread led to an on-farm trial where we basically collected from, I think, 23 different locations in Alabama over a 2-year period. All of the locations are here. We also had a cooperator collect from 10 locations a year in Georgia. And we're basically trying to, again, piece together epidemiology. We trapped aphids. We have CLRDV incidence data. We looked at aphid natural enemies that we captured in the traps, looked at weed hosts around the field, and we're doing an analysis that looks at landscape composition, so like in a GIS type of framework, and also temperature and precipitation variables to try and identify some of the environmental factors and the biotic factors that are really driving this final incidence of CLRDV. Okay, so I'm going to start winding down my talk from here. And I'm going to give you kind of the 5,000-mile view of 3 other studies that I think were important, and they're important to mention. But they're also great examples of why working on brand new pathosystems with not clear methods and not a clear understanding of epidemiology and complex environmental effects is difficult because we have a lot of negative data. So what I'm going to do is I'm going to tell you about the 3 studies that my student Kelly did. I'll take you through some slides, so 3 slides that kind of show you the setup for how we examined nutrient management, planting date, and heat stress, and looked at their impact on CLRDV incidence and yield loss. And then I'm just going to give you the summary slide telling you what we found. So the first thing we looked at was nutrient and CLRDV interactions. We have this wonderful resource at Auburn University that is called the colors rotation. And this is basically a rotation that has 14 different nutrient treatments that have been maintained for over 100 years. So they're basically 8-row plots where you know you will be able to study a nutrient interaction. And you can kind of eliminate the variability that you usually have if you try to study these in a field system. You'll notice that we only have 12 of those treatments listed here. That's because the other 2 are such poor growing conditions, you really don't have any cotton to examine. So we stuck with the 12 we could at least get cotton plants from. In these plots, we basically used the middle 2 rows, and we had a series of experimental steps where we tried to infect certain plants with viruliferative aphids we had in our greenhouse. We tried to exclude aphids for as long as we could off of other plants, basically in an attempt to get healthy and CLRDV infected plants that we could compare. We monitored symptoms on these plants season long. We collected yield data on these plants, and the objective, again, was to look at healthy and infected plants in each of these nutrient deficiencies. Another study that we conducted was looking at age-related resistance. And so in this study, we had 20-foot cages where we basically confined 2 rows of cotton within them. This was done to prevent natural infestations of insects so that we could control when these plants became initially infected with CLRDV. And we would bring in viruliferative aphids and release them in the plant. This is a picture of one of our CLRDV infected plants from the greenhouse. We'd pull off leaves, make sure that all of these plants got infected. And again, we would monitor some of these plants, so 10 plants per plot, for symptoms. We evaluated yield on a per plot basis, but basically had plots that had been infected over 4 successive weeks, beginning at the first true leaf stage. We also had control plots where we did not introduce viruliferative aphids. The hard thing about working with cotton in cages is we had to remove these cages at the end of July, so we couldn't prevent all virus spread that occurred over those 13 weeks. but we could at least delay transmission, like natural transmission, as long as we could. We actually didn't have the results showing that virus spread occurred over those 13 weeks when we were conducting this trial because they were going on concurrently. So we might have done things a little bit different had we known. The third study I wanted to talk about, we basically set up a microplot where we had a screen house with plastic that was stretched over the top to exclude rain. Initially, we were interested in looking at heat and drought, but again, this is a hard pathosystem to work with. You don't always get very high transmission, which is ironic given the high rates of transmission we see in some field locations, but if you try to do it yourself, it's not that easy. And so we never had enough infected plants that we could actually look at the drought treatment. So this basically just became a trial where we looked at plants that were under heat stress. We had weather station in here, and I mean, it got hot enough in here. I think we got up to like 126 degree heat indexes on certain years and days during the summertime. So it was just a very hot environment. We had screen on the inside of the screen house. So this is 100 micron screen, again, to exclude aphids, thrips, all of the pests that we don't want in here to interfere with our experiments. And the goal was to quantify and compare yields between the infected and the healthy plants. Okay, so the take home from these studies. Nutrient deficiencies. We saw that they might influence yield loss under some conditions, but came to the conclusion that other environmental factors are involved, and that's because we saw yield loss in 1 out of 2 years. In year 3, despite our best efforts, we only had 6 CLRDV infected plants across the entire trial, and we tested 80 plants per treatment to try and find infected plants that we could use in our analysis. So CLRDV basically wasn't a problem at this location in the 3rd year. Interestingly, when we did see yield loss in that 1st year, if you average it across all of the treatments, it was about 31% yield loss in the infected plants compared to the plants that were not infected with CLRDV. When we looked at mature plant resistance, we saw yield loss in 1 out of 3 years. And in that year that we saw yield loss, it was the cotton that was the youngest, so that week 1 treatment had a significantly lower yield compared to our control plot. And again, it was about a 25% yield loss in infected plants. The heat stress treatment looked the most promising of all 3 of these trials, and basically we saw yield loss in 2 out of 2 years. We have a 3rd year of data that is ready to be analyzed, but we just got done ginning and weighing yields. So I don't have the 3rd year for you, but in those 2 years, we saw about a 26% yield loss in the infected plants. The other thing that was really interesting, and this was consistent across all 3 of these trials. So if you remember back to my introduction slides where I showed you kind of the doomsday scenario, we had the one field that had complete yield loss. You saw the other slide where there were plants that had a very reduced boll set, and you saw that suite of different symptoms. We didn't really see any visual differences between our healthy and infected plants in these trials. When we analyze things in the nutrient deficiencies, I think the symptoms that come out as significantly different on healthy versus infected plants were the red stems, the red leaves, the red stems, red veins, red petioles. But none of those other symptoms were associated with the infected plants. And again, if you walk through any of these trials, even surprisingly in these microplots where you have healthy and infected plants side by side, you can't tell a difference between them. And so this really begs the question, especially with respect to where we are now with CLRDV, which is the agricultural community doesn't really feel that CLRDV is causing any major losses. And that's because they don't observe any symptoms. They don't observe any signs that anything's wrong. And so these results make me wonder if there could be yield losses occurring, but they're just going undetected because there aren't enough plants in the field for people to question the yield, I guess, the yield difference from what they expected, or just because they don't see anything wrong, they don't know to question it. So that's something that I'm continuing to try to think about, like how do you really explain that to growers, especially if it's not something that they can see? Last thing I think I wanted to end on was, I think from a lot of the research that we've conducted, and folks at UGA have done a good job conducting research on this problem, and I think we have a pretty good understanding of spread, and we know that there's a lot of it out there, especially in certain areas. It doesn't seem to be causing a problem, but moving forward, it might still be a good idea to continue funding for like breeding for host plant resistance. Based on the high number of weeks that the virus is spread during the year, it seems like host plant resistance is going to be the best strategy if this virus ever becomes a problem. So with that, I just wanted to thank you for your time, and I'd be happy to entertain any questions at this time.
So we don't have any questions yet. Just as a reminder, thank you, Alana, for your talk, first of all. It was great, very informative. I appreciate that you took us through those 3 trials to let us know just different things that you found. That was very helpful. Just as a reminder, if you do have a question for Alana, you can put that into the Q&A box at the bottom of your Zoom screen, and we'll make sure those questions get asked. So you were talking about something a second ago. How do you explain something to growers if they can't see it? Do you have any ideas about that, about different ways you can try to explain this to growers, or different things that they can do if they think they might have CLRDV?
Yeah, so I think what I would like to do this year, and this is the best that I've come up with, and if somebody has an idea, I would love to hear it. But I wanted to sit down with an economist and just go through the thought exercise of performing calculations, basically trying to answer the question, so how many, if there's a 25% yield loss on infected plants, how many plants have to realize, how many plants do you have to lose 25% of yield on in order for you to notice there might be a problem? And so that kind of comes down to, the proportion of the plants in the field that are actually affected. And it will involve some calculations based on expected yield and the margin of error around that, which is complicated, right? Because there are different varieties and everything else. But I thought it might be a good place to just start thinking about, how do you try to get at, is this really causing a problem or not?
Yeah, definitely. And it's a different way to show them, right? Like if they can't physically see it, maybe this could be a way to get their attention. I know that.
Right. And it would.
Go ahead.
Yeah, go ahead. Oh, I was just going to say, it basically is a starting point in terms of, if you see something that seems off, like it may be CLRDV, it might not be, but if you don't question it, we won't ever get a handle on it.
Right. And is there a way for them to actually test their plants if they think they might have it?
So we have diagnostic testing procedures for the virus. They're just really expensive. And like I said, so another thing that's complicated, and if you work in cotton, you would recognize this, but a lot of the symptoms, or quote unquote symptoms, that have been found on CLRDV infected plants are also symptoms of other things. So again, unless somebody thinks they have a problem, or unless they're seeing those plants that have bolls dropping or reduced boll set, I don't think it would raise any red flags at this point in time.
That makes sense. Well, I was going to tell you, Alana, I think that some folks at the University of Kentucky took that economic approach for, it was either pests or diseases in corn. I can't remember which one specifically, but I'd be glad to get you in contact with them if you want ideas for how to use an economist for that.
That would be great.
And well,
Thank you.
Yeah, of course. And I'm not seeing any more questions right now. But if you were wanting to leave the attendees with just a takeaway for today, what would that takeaway be?
So I think the takeaway is the CLRDV doesn't seem to be an economic problem at this point in time. There aren't obvious widespread losses, but we might be experiencing some yield loss from CLRDV that goes undetected. And we're still trying to figure out those environmental factors that could be underlying it. So kind of stay tuned if it's something that's on your radar.
Well, thank you again, Alana, for the presentation today. Again, Alana Jacobson at Auburn University. Just as a reminder, a link to the recording of this will be sent to your email and available on our website after the webinar. And if you ever want any more information about the Southern IPM Center, you can visit southernipm.org. So thank you again, Alana, and we'll see you next time.
Great, thank you.